Good morning, everyone.
Morning.
Okay, welcome back, everybody. Hope you're doing well. And it's nice that the days are getting a little warmer this week, right? Last week was a little bit chilly in some ways. So I hope you and your families continue to be safe and healthy. Believe it or not, we have lecture exam 2 next Tuesday, right? A week from today, you'll be taking lecture exam 2 on chapters 5, 6, 9, and 10. Okay, so there's lots to do here. We're going to try our best to cover most of chapter 10 today to finish that off to get you into a good position for your exam. And expect on Thursday, even though I intend to start chapter 12 on Thursday, I'll also give class time to talk about the exam, give you an opportunity to ask questions about content that you might be concerned with on the exam, so you'll get a chance to do that. We also have lab this afternoon, of course, or later this morning, I guess I should say, and we will be starting the nervous system content, which represents the final 1/3 of our semester together. Okay, so things are starting to move towards that end point, folks. Do you have any questions you'd like to ask right now? Okay, all right, well, please let me know if you do. Remember, office hours are available on your syllabus Monday through Thursday. Just pop in and see me if you need something, okay? All right, so we're in chapter 10. Last time we spent a fair amount of time describing some of the molecular anatomy of muscle cells, you know, the sarcoplasmic reticulum, the myofibrils, all that good stuff. We finished our notes last time talking about some important muscle proteins such as actin and myosin, titin, dystrophin, troponin, tropomyosin. These things are all in your notes from last class. So I'm not going to continue with that description. I'm going to start some new ideas here today. But to get our brains thinking about the physiology of muscle contraction, what I'm going to do is start with a topic where we try to connect the idea of muscle contraction to the subsequent motion of a bone. Okay, so let's see if we can design a flow chart which describes this. I'm going to share my screen with you. Okay, and bring up our note-taking document. All right. Okay, so our heading this morning that we're going to start with is the pathway between muscle contraction and bone movement. And we're just going to design a flow chart here that attempts to describe this idea. Okay. So the very first thing that's going to happen when muscles contract is the anatomy inside the muscle fiber has to shorten. So we could start that at a number of different places, but I think the most logical starting place is to talk about the sarcomere. Remember, the sarcomere is a contractile unit of a myofibril. If you need to be remembered what a sarcomere looks like, just look at figure 10.2, and take a look at panel D as in David. Panel D as in David, where the figure legend for panel D says a simplistic representation of the components of a muscle fiber. Have a look at that and you will see sarcomere in the middle of that image. Okay. So the very first thing is the sarcomere will shorten. Now, of course, you could say, well, what causes the sarcomere to shorten? We are going to handle that today. We are going to describe that today. And the honest answer is that the sarcomere is going to shorten in response to some events that are initiated by the nervous system, okay? But we're going to start here. The sarcomere shortens because the sarcomere is a structural subunit of the larger myofibril. Again, I reference figure 10.2, panel D, because the sarcomere shortens, the myofibril is going to shorten. When the myofibril is shortened, it causes the actual muscle cell to shorten. Now let's talk about how that's possible. We finished last class talking about a special protein called dystrophin. Again, put your eyeball on dystrophin, figure 10.2, picture D there. So when the myofibril shortens, you can see the myofibril is going to tug on the dystrophin, and the dystrophin is connected to the inner surface of the sarcolemma, which is the plasma membrane of the muscle cell itself. So the myofibril shortens, this tugs on the dystrophin protein. And because the dystrophin protein is attached to the inner surface of the sarcolemma, this is going to tug on the sarcolemma. Okay, remember the analogy we talked about last time when we were talking about fascia, where we had your t-shirt stitched to your sweatshirt, that kind of thing. If you pull on one, everything else moves. That's the idea we're using right now. So when the sarcomere shortens, it shortens the myofibril. When the myofibril shortens, well, it pulls on the dystrophin protein, which pulls on the sarcolemma, which basically means the entire muscle cell is going to shorten. Okay. All right, now we're not quite finished yet. Okay. So let me ask a question in the chat window. Okay. Is the flow chart that we're talking about making sense so far? You have some familiarity with these words. You can understand when one thing shortens, the next thing shortens, the next thing shortens. Are you okay? Any questions you'd like to ask? Okay, so let's keep rolling then. All right, so we've reached a point in our flow chart where the entire muscle cell is shortening because of events that started with the sarcomere shortening. Okay, now you know, because we talked about it last time, that skeletal muscle is going to have 3 different layers of fascia wrapped around it at different levels of structural organization. So number 5, the entire muscle cell shortens. When the muscle cell shortens, that's going to tug on the layer of fascia that's around individual muscle cells, which is the endomysium. So when the entire muscle cell shortens, it's going to tug on the endomysium. Okay, that's the fascia around individual muscle fibers. Because the endomysium is physically continuous with the next layer of fascia, which wraps around groups of muscle cells called fascicles, that next layer of fascia is going to get tugged on. That next layer of fascia is called the paramecium. Because the paramecium is physically continuous with the third and outermost layer of fascia that wraps around the entire muscle itself, that fascia is going to get tugged on. The name of that fascia is the epimysium. Okay, we mentioned to you that these 3 layers of muscle fascia are physically continuous with the tendon. So when you tug on the epimysium, it's going to transmit that tension to the tendon. Okay, so number 9, we tug on the tendon. And number 10, when you tug on the tendon, of course, we're getting towards the bone, but we're going to be, a bit more anal here. When you tug on the tendon, you have to ask yourself, well, in what manner is the tendon attached to the bone? Well, the tendon is going to be stitched to the periosteum. Okay, so when you tug on the tendon, all of a sudden you start tugging, you start pulling on the periosteum, which is that membrane we've described previously that covers the bone. And of course, we know that the periosteum, which covers the bone, is actually physically stitched via Chpt fibers, right? There's bundles of collagen fibers to the surface of the compact bone. And when you tug on the periosteum, you tug on the compact bone, you tug on the bone itself, you move the bone. The bone actually moves, folks. Okay, so one more time, we shorten the sarcomere, which then causes the myofibril to shorten, which then tugs on the dystrophin protein, which tugs on the plasma membrane of the muscle cell called the sarcolemma. That causes the muscle cell itself to shorten. The muscle cell is wrapped in fascia, so that causes the endomysium to get tugged on, which then tugs on the paramecium, which tugs on the epimecium around the entire muscle, which tugs on the tendon, which tugs on the periosteum covering the bone, which tugs on the bone, and finally the bone physically moves, right? It is amazing how complicated this stuff really is, right? And it's also an amazing piece of biological engineering that Mother Nature has designed and come up with these solutions over arguably millions of years of evolution. Okay. All right, folks. So any questions about our flow chart? Okay, I think we're in a good place now to start talking about muscle contraction. And as I look through your textbook, it's clear to me that the authors that wrote your book have a certain way they want to present information. Just as an example, if you take a look at figures 10.5 and figures 10.6, right, they're showing you information about the physiology of muscle contraction. And then as we move deeper into the chapter, deeper into the chapter beyond those topics, you get to other important figures as well, including figure 10.7, 10.8, and 10.9. Okay, 10.10. There's a lot of important figures here. So the textbook is presenting muscle physiology first by explaining the actual process of muscle contraction at the level of the muscle cells themselves, and then goes on to explain how the nervous system triggers these events. Okay, so from my perspective, I would prefer to talk about the nervous system first, which is actually the trigger for the events, and then talk about the muscle physiology of contraction itself. So I'm going to cover this in a slightly different order than your textbook. There's nothing wrong with your textbook. I just prefer to describe these events in the actual way it's really going to happen, okay? So to do that, so we're going to have a description, of events at the neuromuscular junction, because it's a description of events at the neuromuscular junction, which is responsible for triggering the actual muscle contraction itself. Okay, so folks, I'm going to show you a Wiley Plus video right now. Okay, and so I'm not gonna it's not like I'm not showing you a YouTube video, which I would typically put a link into your notes. but I'm going to show you a Wiley Plus video and you all have access to it, remember? So let me just remind you of some things, right? If you're in Wiley Plus and you go to Wiley Course Resources, you'll come up with the course-wide resources and go to 3D Physiology Animations. Here, I'm going to bring them up next. Hold on a sec. Here's the animations. I'm going to start this morning by showing you the neuromuscular junction video. The neuromuscular junction video, okay? So don't forget about this. Inside the 3D physiology videos, neuromuscular junction video, okay? So we're going to show it, then we're going to talk about it, okay? All right, gang, here we go. It's only about 2.5 minute video. Let's see what it has to teach us. Here we go.
Movement in the body is caused by skeletal muscle contraction, which is muscle shortening. Nerve impulses originating from the brain or spinal cord excite skeletal muscle cells to initiate muscle contraction. A group of muscle cells stimulated by a single motor neuron is known as a motor unit. There can be many motor units in a whole muscle. Each muscle cell consists of many rod-like myofibrils that contain the contractile proteins of the cell. To cause muscle contraction, the motor neuron must initiate an electrical impulse in the skeletal muscle cells, even though the muscle and neuron cells do not touch. There is a gap known as the synaptic cleft between the neuron membrane and the muscle cell membrane that only a chemical signal can cross. The neuromuscular junction is this chemical synapse between the motor neuron membrane and the muscle cell membrane. Once the electrical impulse arrives at the end of the motor neuron, voltage-dependent calcium ion channels open, thereby allowing calcium to enter the neuron. This increase in intracellular calcium causes the release of neurotransmitter acetylcholine into the synaptic cleft. At the muscle cell membrane, known as the sarcolemma, the neurotransmitter attaches to chemically gated sodium channels, causing the channels to open. This allows the passage of sodium into the skeletal muscle cell. The entry of sodium creates an electrical impulse, also known as an action potential, which excites the sarcolemma. Once an action potential is propagated along the sarcolemma, any remaining acetylcholine in the synaptic cleft is broken down by the enzyme acetylcholinesterase. At this point, action potentials will stop being produced by the sarcolemma and excitation of skeletal muscle cells will end.
Okay, folks, so there we have a quick little video describing the events at the neuromuscular junction. We need to talk about this. You know, this is one of the major essays on your upcoming exam in 1 week. Okay, so we're going to spend some time on this, and then we'll also move into the physiology of the muscle contraction itself. Do you have any questions from having watched the video one time? So I appreciate that it can be a little intimidating, like many things in this course, because it's so darn complicated. There's so many different things going on. Our goal today is to decomplicate this for you. Okay. All right. So I'm gonna do some drawing here because it's just easier for me to explain it by drawing than it is typing words. And I would turn your attention please, to figure 10.9, and have that available to look at during our conversation here this morning. Okay, so let's continue here. And as I said, I'm going to do a bit of drawing. All right, and okay, I guess I'll use a dark blue to start out with. Okay, so what you'll want to do is take a look at my drawing, which will not be the prettiest because I just don't have artistic ability, and match it up with ideas and pieces of artwork that you see in figure 10.9. Okay. All right. So, we now know a little bit about the anatomy of a muscle cell. We really haven't talked too much about, well, how does the nervous system trigger contraction of the muscle cell? That's really what we're talking about at this moment. How does the nervous system trigger contraction of a muscle cell? Okay, so we're going to start out by drawing a neuron, and I'm going to draw part of the neuron. Okay, and so what we have here is an axon. Okay, remember, an axon is part of a neuron. And we also have the muscle cell itself. This is going to be the sarcolemma. I encourage you to draw at your house, because by drawing, you're forcing your brain to focus on content in the moment, which helps it stick. Okay, so we have an axon here and a muscle cell. So this way would be inside the muscle cell. Okay. All right. So you might ask yourself, well, what the heck are we really looking at here? Okay, if we think about everything we've drawn collectively, the neuron as it approaches the muscle fiber itself, okay, this is the neuromuscular junction. Okay, so when we talk about the neuron and its physical proximity to the muscle cell, both of those things coming together, that's the neuromuscular junction. Okay, we also have this space here that is in between the neuron and the muscle cell. This is known as the synaptic cleft. Okay. This little bulb-like structure at the end of the axon is known by different names. In your book, I'm seeing the synaptic end bulb as the phrase your book is using. The synaptic end bulb is also known as the synaptic knob in some textbooks. And I've even also heard it called the terminal bouton. So synaptic end bulb, synaptic knob, terminal bouton are all the same structures, folks, all the same structure, okay? All right. So for your nervous system to stimulate muscle contraction, the nervous system is going to send an electrical signal down the neuron. For example, if you say to yourself, if your own mind is saying to your body, okay, lift my left arm up. Okay, for that to happen, your mind is controlling the events. Your mind, which is a product of the biochemistry of your brain, is generating electrical signals that then propagate down to the muscles of your shoulder and arm so that you can lift the arm up. Okay, so all of this is going to be regulated and controlled by electrical signals. Okay, so I'm going to try to take some notes here that very roughly tell you what's going to happen. So number one, an electric signal moves down the axon. And you ask yourself, well, what created the electrical signal? Hold on to that question, because it's not something we can talk about at this moment. We will be talking about it when we get to the nervous system chapter next. Chapter 12 starts nervous system, okay? So your brain, which is the warehouse of your mind, generates an electrical signal. It moves down the axon. When we're talking about an electric signal, that electrical signal is properly called an action potential or a nerve impulse. Basically, what we have is, and for right now, I'm just going to skip a little bit of detail so I can move the conversation along. But the electrical signal is basically the motion of ions, right? Remember that term ion, a charged atom? The electrical signal is the motion of ions out of the cell and back into the cell, out of the cell, back into the cell. So we have this zigzag pattern of ions moving back and forth across the membrane of the neuron. This is the actual electrical signal. An electrical signal in biological systems like your body always represents a motion of ions. Okay, so the green here, the green is, and I can do it on this side as well. The green is the action potential. Okay, so the electrical signal at note number one is the action potential, and it moves down the axon. And it was created because you had a thought that you wanted to lift your left arm up. Okay, you thought it, and your body responded by generating an electrical impulse. Okay, so that electrical signal is going to spread down the it's going to spread down the plasma membrane of the neuron, the neurolemma. Okay, and it's going to encounter special channels that are embedded in the membrane. And so I'm going to draw these as orange little rectangles. And these orange little rectangles are voltage-gated calcium channels. Okay, so I bet you've heard the word volt at some moment of your life, or voltage, right? Maybe some of you have a little bit of experience with these sort of topics. You know, it's not uncommon for people to do a little bit of basic wiring in their home, like, you know, hooking up a new light fixture in the kitchen or something like this. At any rate, we at least need to talk a little bit about voltage. Okay, so let me ask you in the chat window, can you give me a definition for voltage? What is voltage? What is voltage? Anybody? It's kind of one of those odd things you think to yourself, well, why do I have to be an electrical engineer when I'm in a biology course? But the reality of it is, much of your physiology relies on electrical principles. Okay, thanks, Mark, for contributing. Mark says current of energy, and I think I can get there with what you're saying. I'm going to define it differently for the purposes of our conversation, but I appreciate the contribution. So remember, in biological systems, the electrical signal is the motion of ions, right? It is the motion of ions. Well, voltage is the separation of charge across the membrane. In this case, it's going to be the separation of charge across the plasma membrane of the neuron. Okay, so what I'm saying is when the electrical signal, which is that green action potential in our drawing, when it reaches these voltage-gated calcium channels, the ions that are the essence of the electrical signal, well, some of these ions bind to the voltage-gated calcium channels and change the shape of the channel. And when it changes the shape, the channel actually opens up. Okay, so let me ask you this in the chat window. What are the voltage-gated calcium channels made of? What are they made of? Do you know? Let me ask you this. Is the voltage-gated calcium channel a carbohydrate, a lipid, a protein, or a nucleic acid? Okay, thank you, Sierra. It's a good guess. You're absolutely right. Voltage-gated calcium channels are proteins. So stay with me, folks. Stay with me. Here's another question. What are proteins made of? What are proteins made of? All right, this is where your basic beginning biology knowledge becomes critical. What are proteins made of, folks? Come on. I know it's rumbling around in there. Help me out. Okay, thank you, Christina and Amy. You're absolutely right. Proteins are made of amino acids. Good job. So the voltage-gated calcium channels are actually proteins, and we remember that proteins are made of amino acids. What you may or may not remember is that amino acids can sometimes take on an electric charge. They can become electrically charged. If you don't believe me, take a look at figure 2.20 in your textbook. Take a look at figure 2.20 in your textbook. Take a look at figure 2.20 in your textbook. Okay, I'm going to ask you a question. Here's a question. In figure 2.20 in your textbook, part B, what charge does the pink shaded group of atoms have? What charge does the pink shaded group of atoms have? Figure 2.20b, part B, what charge does the pink shaded group of atoms have? Okay. Do you see it, folks? Part B at the top right of figure 2.20, you should see a group of atoms which are colored pink, and there's an electric charge represented there. What is that charge? Okay, all right. Amy says it's negatively charged, and she's absolutely right. In other words, there's an oxygen atom there that has a negative charge. So let me ask you, do you see the negatively charged oxygen atom of figure 2.20 part B. Come on, folks, let's get some responses here. I'm not continuing until I get more participation. Let's go. All right, good, good. We have a negative charge on that oxygen. And let me ask another question. What atom has a positive charge in that same picture? What atom has a positive charge in that same picture? Interesting. I'm looking at the responses in the chat window, and I'm not getting correct answers from you. Okay, so something's not right. Okay, folks, let me give you the correct answer and see if you can find it. Okay, don't you see a nitrogen with a little plus charge? A nitrogen, which is a little N symbol with a plus charge. Okay, do you see that? Okay, all right. So, all right. So thank you for doing that, right? I need you to engage the content. If you're not engaging, you're not learning. Okay. So my point here is that the voltage-gated calcium channels are made of protein. Proteins are made of amino acids, and amino acids, such as those you see in figure 2.20, at different moments of time, can themselves have an electric charge. So what I'm telling you is the ions of the action potential have the ability to bind to some of the amino acids making up the structure of the voltage-gated calcium channel. The ions of the action potential bind to the proteins that are the voltage-gated calcium channels, change their shape in such a way that they open, the channel opens. Okay, and so let's see here. I'm going to quickly run out of space here, so I'm sorry about that, but let me do the best I can to squeak in a few little notes. Okay. I'll come down here for number 2. Okay. Ions of the action potential. Ions of the action potential bind to the voltage-gated calcium channels, VGCCs, and change their shape. When they change the shape, the channel opens. You say, well, what does it open to? Okay. So in the synaptic cleft, you're going to have synaptic fluid, right? So in the synaptic cleft, you have synaptic fluid. You know, I'm not going to draw it because, you know, how do you draw fluid? But please understand the synaptic cleft is not an open space. It's simply an area where there's fluid. So the synaptic cleft has synaptic fluid. And what you may or may not be aware of is that in the synaptic cleft of the synaptic fluid, you're going to have calcium ions. So here in the synaptic fluid are calcium ions, Ca+2. When the action potential comes down the neuron, the ions of the action potential bind to the proteins of the voltage-gated calcium channels, changes their shape, opens the channel so that these tiny little calcium ions actually go through the channel and enter the neuron. Okay, so we'll come down here and say channel opens and calcium ions enter. Calcium ions enter. Okay. Questions right now? Okay, so if you're following along, again, I'm back, and personally, I'm back looking at figure 10.9 now in your textbook. Let's continue. And so let me ask some of you folks, let me ask a question here. Okay, so I just want to